Unit 7 Notes video. We're going to be learning about genetics, basically the beginning of genetics, focusing our attention on one of the most famous scientists of all time, Gregor Mendel, who was around in the 1800s, and the reason that's important is because that was also during the time of Charles Darwin, which we'll talk about towards the end of the year. So there he is, Gregor Mendel, and not dates that you'll have to know, but again, significant because of basically the theory of evolution as Charles Darwin came up with it in the 1860s kind of shocked the scientific world, and so Mendel's genetics kind of took a back seat. So anyways, he was an Austrian monk. He's known as the father of modern genetics. Not that this is important, but it's related because basically monks have a lot of time on their hands, and so when he's breeding pea plants, you can imagine the time he had to get into very specific details and carry out pretty tremendous work. What he found was basically inheritance of traits in pea plants that later became laws, and they are known as Mendel's laws of inheritance. The first thing we'll talk about is heredity, and this is a pretty simple definition. It's the passing on of characteristics from parent to offspring, and this is obviously not just pea plants, but for people like you too. Inherited characteristics are called traits, and we kind of already talked about this when we started talking about meiosis. Mendel called them factors. Again, this is the 1800s. He had no really idea what DNA was, how it worked, and all the details that we have talked about and will talk about. So he was not because of anything besides technology did he not know these things. And so the branch of biology that studies heredity is called genetics, and since this time it's become one of the largest fields of biology, and definitely this prominent figure kind of started it all, and it was founded upon as you can see there, the rediscovery of his work. It was not as significant at the time to people that were more in tune to what Charles Darwin was saying about evolution of humans especially. So here you can see the data for Mendel's pea plants. He had about 28,000 plants, and it doesn't take a real scientist to understand that lots of data means pretty good numbers, and probably going to represent the population pretty well. And so again, we're going to focus on pea plants. That's all he did, most of what he did. And pea plants reproduce sexually, just like people do, making male and female gametes, sex cells, just like we talked about in meiosis. The male gamete forms in pollen, and the female gamete formed in the ovary. And fertilization, like we've already talked about, occurs when the male gamete unites with the female gamete. So pollen, or parts from the pollen, the sperm, are forming with an egg. And again, these are plant parts, but very similar to how it works for you and I. Pollination is the actual vocab term that refers to the transfer of pollen to ovary in a plant. This is how normal reproduction takes place, and you can see some plant parts there. So what Mendel did was he removed some pea plant parts actually in the flower to prevent these flowers from self-pollinating. And all that means is that the pollen from the same plant pollinates its own ovary, so it's basically having sex with itself. And so here's the flower parts. Not that you'll have to know this for the test or anything, but it will be helpful to understand how this all works. This picture can be found in your book. You can see a similar picture on page 642. So the pistil here is basically a big long tube, and at the bottom of that tube is this area where the ovary contains the egg that can be fertilized by the pollen that is produced on these stamen. So right up here at the top are the pollen. You can see they're kind of yellow colored, and typically that's stuff we think about when we think about bees going around from flower to flower and gathering all that stuff on their legs, right? So they're just a typical flower picture, and just so you kind of see how this works, the pollen that is up here at the top of the stamen would go into the pistil down in this tube, and then eventually would pollinate the egg and produce more plants. This is a little bit different than what you have, and I just cut down on some of the pictures, but as long as you look at it once, I think it'll make sense. Cross-pollination is a pretty important concept for this unit to understand, again, how Mendel did this work. Simply transferring pollen of one plant to the ovary of another plant. And there's a picture down there, you can see kind of what that's talking about. And he did this to make sure that he was breeding certain plants with each other. He didn't want them to just sit out in the field and breed however they wanted to. He wanted to control everything in order to get good scientific data. Controlling how plants breed is going to be an important part of that. And to say that he was very careful with all of his work is pretty much a huge understatement. Again, 28,000 pea plants. As a monk, there's really not much else to do, so maybe meditate and stuff, but that's about it. But basically, here you go. You remove the male parts from the flower, leaving only the female parts left. And then from another flower, he could take the male parts, pollen, on a paintbrush, and just simply brush that paintbrush right onto the pistil here, and cause these plants to pollinate with each other. That's cross-pollination. So he was able to create a whole generations of tall plants and whole generations of short plants that he called purebreds. In other words, no matter how many times these tall plants mated with each other, they created tall offspring. That's purebred. And same things for these short plants. No matter how many times they mated with each other, they created short offspring. So he referred to these offspring of purebred tall and purebred short as hybrids. And so here's a picture. Right here is a purebred short and a purebred tall. When they cross, you get all hybrid plants. And so what he did is actually crossed 6-foot tall plants with 2-foot tall plants. And the result, right here, is 100% 6-foot tall plants. Naturally, you would think of maybe 3 or 4-foot tall plants taking place, but it didn't happen. The first generation here is called the P1, or parental generation. Second generation, or first offspring generation, is called F1. The F refers to filial. Not that really helps because it doesn't really ring a bell with anyone, but that's what the F stands for. And then here's the F2. And what happens is after you cross these hybrid offspring, these guys right here, that were 100% tall, you get a very strange and consistent, most importantly, ratio of 3 to 1. 3 tall to 1 short. And this is very easy to see in Mendel's work, and we'll take a look at this at the end of the slideshow. So what does monohybrid refer to? Basically, we're talking about information from this table right here. It's all dominant and recessive traits from pea plants. We'll get to that in just a second. But basically, when Mendel crossed a purebred tall with a purebred short, he got all tall plants, 100% tall plants. You would think that he'd get some kind of medium or in-between height. But then again, when he crossed those purebred, again, purple flower, with a purebred white flower, again opposite, you would think somewhere in between, maybe polka dots or something, but no, you get all purple flowers. Okay, and so he referred to this trait that was observed. This is the key here. It was the trait that was observed in the offspring. He referred to that as dominant. Okay, the trait that was observed is dominant. The trait that seemed to disappear, in other words, this purebred tall and purebred short that created all tall plants, so the short seemed to disappear. So he called the short recessive. Same thing for this case with the purple flowers. The white flower would have been the recessive because it seemed to disappear in that F1 generation, the first offspring. So Mendel concluded that these plants have factors. Again, he didn't know what DNA and chromosomes and all the stuff that we know were all about. But he said that these factors control the traits, such as color, shape, and height. Now we call these factors genes, just small segments of DNA that code for proteins. And alternative forms of these genes, like we talked about already in meiosis, are known as alleles. In other words, the gene for height, but the alleles are tall or short. The gene for color of the pod, for example, but the alleles are yellow versus green. And so here's a table that you should know kind of the difference between dominant and recessive. You should be familiar with all these by the time we get to test time, but especially the colors of the seeds, shapes of the seeds, colors of the flowers, okay, pod shape and pod color, tall plant versus short plant. You should know what all these are. You're not really going to see these. These would be kind of too confusing to notice on a test. But all these other ones, you should be able to recognize which one is dominant and which one is recessive. And you're going to do a lot of work with them, so hopefully that helps. Oh, and answers for your previous fill in the blanks in case you weren't sure what I was saying. Here's dominant, And here's the word recessive that I was talking about, the trait that appears and the trait that seems to disappear. So these two alleles, like we kind of started talking about in meiosis, for each trait can be expressed as a single letter. And for plant height, we'll just use the letter T. You could use any letter. It could be A, B, C, doesn't really matter. The dominant allele, we're going to use capital T. Okay, dominant, capital letter, kind of relate. And then recessive will obviously use the lowercase t. And hopefully you're starting to get some recall from middle school when you guys first did this. Mendel's purebred tall plants, therefore, were big T. His purebred recessive plants were obviously then little t. So we're going to fill in the blanks to the right. In the picture, here's a purebred tall plant, purebred short plant. And so obviously the tall plant had to have big T, and since it's purebred, two are the same. And then the short plant, obviously the opposite. But again, purebred, so it has two of the same. Okay, that's going to be important for later on for vocab and for filling out worksheets, and then obviously getting ready to the test. The F1 generation is going to be one of each. Big T from this parent and little t from this parent. Okay, again, hybrid, but this offspring looks tall. It looks like whatever it got from this plant parent, which was big T, little t, kind of seems to disappear. So, Mendel came up with the conclusion that the allele, that form of the gene, for tall plants was dominant to the allele for short plants, just because of this picture here. Because he saw tall when he mixed purebred tall with purebred short. He saw tall in the offspring. Confirms that the plants had two alleles, or two factors, for each trait. And here are the letters. Knowing that the traits are inherited from the parents, he also concluded that these alleles are inherited. Again, he didn't know about meiosis, DNA, genes. He didn't know about any of those things. I mean, he knew that stuff was in there, or something was in there causing it to happen, but he didn't know the details that we know today. So, something else that he came up with that was pretty obvious to us now, the plant can only get one allele from each parent. Okay, and we saw this in meiosis. The gametes contained either one of those forms of the factors. Again, he called them factors, we call them genes, and we're going to refer to them as big T little t, big A little a, big B little b, all the time just using letters. Right? And again, this goes right back to meiosis. This refers to basically Mendel, one of the Mendel's laws that he came up with from his work, and that is the law of segregation. Okay, segregation, just like segregation in America of blacks versus whites. It's the same exact idea. Okay, these things separate. Okay, so segregation states that every individual, every organism has two alleles of each gene. And when gametes are produced, and again we saw this in meiosis, each gamete receives one of these alleles. Again, something we already know. Half of your DNA comes from mom, half comes from dad. But at some point, these genetics had to get figured out. So, some vocab words that we're going to wrap up this with. Phenotype, and again, focus on the first two letters here. It's going to help. Phenotype refers to the organism's physical characteristics. Again, physical characteristics. You actually see a tall plant. Good example. Alright, you look at the person next to you in class, you see brown hair, you see brown eyes, you see their physical characteristics. That's phenotype. The next one is genotype, and these are the opposite. Okay, genotype, again G-E-N, because it refers to the organism's genetic makeup. When you turn to someone in class, you're not looking at their genes, their chromosomes. You're looking at tall or short, or brown hair, or light eyes, or whatever. That's the genetic makeup, is the genotype. It's what you can't physically see. So again, we're going to represent those with just letters. Homozygous, or purebred, represents the two alleles that are the same. Homozygous or purebred, they're used interchangeably. And heterozygous or hybrid refers to different. If you think of a hybrid car, okay, it uses energy from gasoline and from electricity, has different alleles, big T and a little t. How are you going to distinguish between homo and hetero? I think we all know the answer to that.